they are filterable? They're really tiny and they go through a filter. That's all that means. So really tiny is kind of a relative thing that you have to be used to thinking about. So if everybody's here, there's 43 people in the room. And so they tell how many cookers don't work by subtracting the number up there you get. Everybody try to vote. Okay. Whoa. What's the likelihood of that? So 15% said they do not have nucleic acid. 21 said they're obligate intracellular parasites. C, 30% said they're filterable. D said they're not composed of cells. Viruses differ from bacteria in that viruses are bacteria composed of cells. They are cells. Yes, they're single cellular cell organisms. Bacteria are cells. Viruses are not composed of cells. What's the basic unit of a virus? A virion is the basic unit of a virus. Basic unit of a bacterium and all living things is a cell. The thing about filterable, that's kind of an annoying question in there. Filterable means they're really small, and some bacteria are also really small. Some viruses are really huge. We'll talk in a second about the range and sizes of viruses, but that's not the answer. Our obligate intracellular parasites. Some bacteria are obligate intracellular parasites. Some kinds of bacteria actually have to live inside your cells. We'll talk about that later. All viruses have to live inside your cells. Do not have any nucleic acid. They all have nucleic acids. RNA and DNA are the nucleic acids. They're polymers of nucleic acids. So the correct answer is D. Viruses are not composed of cells, while bacteria are composed of cells. All right, let's try again. Whoops. Which of the following statements about spikes is false? They're used for absorption. They're used for nucleic acid replication. They're found only on envelope-viruses. They may cause hemagglutination. So hemagglutination refers to the clumping of red blood cells together based on carbohydrate surface on the surface of the red blood cells. So which of the following is false about spikes? The following is false. See, false is even in big capital letters. So okay. Oh boy. Most you were split. Both answer A and answer C said that this is false. They're used for absorption. C, they're found only in envelope viruses. The next answer was B, they're used for nucleic acid replication. D, they may cause hemagglutination. You guys talk to your neighbor for a moment. Talk about the answer you put and the answer your neighbor put, and then we're going to vote again. And I'll tell you, D is true. They may cause hemagglutination because they cause cells to stick together. We'll talk about why that is in a second. Did you see what's Okay, guys, let's do overtime. Time for a do-over. Vote again. Or you're voting faster. This question? Or you're just leaving your vote? I mean, okay. So I think we're moving in the right direction here. I told you D wasn't one, so at least you put that in. So why do people put C? They're found only in envelope viruses. Somebody asked a question about that. Somebody over here, when you're talking about, was it you, Amanda? You asked, what about viruses that don't have an envelope? And we looked at some examples of icosahedral or polyhedral viruses. And there's a little ugly electron micrograph in here that shows that some of them do have spikes, for sure. They just, the unfortunate thing is the figures in the book that they use for teaching about spikes happen to be an envelope virus. But you don't have to have an envelope to have a spike. So that takes us to A and B. Are they used for, so where are the spikes found? Well, they're found around the outside of the capsid. If there's a membrane, they'd be in the membrane, but they're on the outside. They're certainly not inside the capsid where the nucleic acid is. In fact, once the nucleic acid gets replicated, where does that happen, nucleic acid replication? It happens in the host cell, and by that time the spikes are kind of gone and irrelevant, right? So they're not involved in nucleic acid replication. They're used for absorption. You know what I realized after I put this up here? This is a slide that came with this chapter in the book. And what is absorption? It has something to do with sponges and sucking stuff up. I don't know. The word that you should say is adsorption or adherence. Adsorption or adherence are the words we use. Tell me that's why you guys all got this wrong on the first try, right? It was really because the book screwed this up. It wasn't you, it was them. They're used for adsorption or sticking to the surface of the cell. And remember, they're really specific. The spikes have a part of them on the end that recognizes the structure on the surface of the cell, which might be a carbohydrate, a sugar, some kind of glycosidic linkage out there, and the spike is really specific to that, and that's what controls what kind of cell gets infected by what kind of virus. That's what controls why birds get infected with bird flu so much easier than we do. We'll talk about that more when we talk about epidemiology and influenza and that kind of stuff. Questions about spikes? Okay. Viruses range in size. So we just talked about size a second ago when we were talking about filterable, right? Viruses range in size from 10 to 100 nanometers in length, 10 to 1,000 micrometers in length, 2 to 100 nanometers, 20 to 1,000 nanometers. So remember how big a nanometer is? So I can kind of picture, after having been a microbiologist for 20 years, how big a micron is, even though you can't really see a micron. The nanometers, 1,000 times smaller than a micrometer, which is kind of outside the range of what I can imagine you appreciate personally, but that doesn't excuse me from the responsibility to know that. Okay, I'm going to close it. 5, 4, 3, 2, 1. Oh my God. All right, I'm going to tell you something. We're going to talk to your neighbor and do over. If you change the units on micrometers, what would 1,000 micrometers be? Millimeter. You know how big a millimeter is? It's visible. So it's not B. So talk to your neighbor and try again. It's not B. Alright, we're closing in on it. Half of the people said 10 to 100 nanometers, half of the people said 20 to 1,000 nanometers. 1,000 nanometers is the same as a micrometer. And in fact, some of, let's look at this video. Some of the viruses people now know. Some of them, and this isn't even included in this book. In the last couple of years, people have discovered viruses that are even bigger than these. These are all viruses up here. These are all viruses. This is a red blood cell. This is E. coli. It's about 3 micrometers long and 1 micrometer across. That's about the normal size of bacteria you're going to see in the lab this year. The cocci that you're going to look at are more like a micron in diameter, maybe a little bit more. The viruses are tiny. Here's one that's 24 nanometers. Ebola virus, which you hear a lot about right now, is huge, and it's almost a micrometer. So it's about 1,000 nanometers. So, and there are actually some viruses that have been discovered recently that live in the environment and infect amoeba that are even bigger than that. And we don't really know what the deal with those is yet. So the correct answer is D. Alright, I think we got one more. Maybe 2 more. Viruses cannot be cultured in. What does it take to culture viruses? So I could just tell you, but I want you to think about this for a moment. I have already told you. Viruses cannot be cultured in laboratory animals, in cell culture, in embryonated eggs, or in culture media. Culture media is like the broth you grow stuff in the lab. So it's kind of like culture media. You guys know what cell culture is? Cell culture is when eukaryotic cells are grown in a petri dish. You can take cells out of your blood, or cells out of a tumor, or cells out of all kinds of different tissues, divide them up, put them in culture media, and grow them artificially in a petri dish. Okay, 5 seconds. They cannot be cultured in culture media. You guys got that right. Whoops, where'd that go? Sorry about that. They cannot be cultured in culture media. Accidentally erased it. Remember we talked about they can be grown in eggs. That's how influenza virus gets made for making vaccine. They can sure be cultured in culture cells in laboratory animals. We're an animal, right? Of course we can culture an egg. Okay, one more. Which type of viral nucleic acid functions as mRNA? Double-stranded RNA, negative strand RNA, positive strand RNA, RNA reverse transcriptase. You guys are voting fast on this. C. Almost everybody put C. That's good. The positive strand is the coding strand. The positive strand is mRNA. Nobody put this. This is the one that you throw in just to make sure that people are reading it. Reverse transcriptase is an enzyme. There's no, that doesn't even make sense, right? What's reverse transcriptase for? Can we talk about this? It's for retroviruses. So for viruses that enter the cell as RNA and be converted back into DNA to stay in the host cell's genome, that's what reverse transcriptase is associated with. Okay, any questions or comments about any of these? We are going to switch back to the section on bacterial structure. When we ended last time, we were talking about flagella. And we said that they are the whip-like propeller, I should say, the whip-like propeller for locomotion. We talked about the parts. The filament is the long part. The hook connects to the basal body. The basal body anchors the cells. The flagellum to the cell. We said you guys are going to know the arrangement. So So Libby and I, strangely, between the two of us, have a collection of stuffed microorganisms. And these are the flagella of this guy. My wife thinks this one looks like me, because it's only got 6 hairs left on it. What would you say about the arrangement of the flagella around this? What category would these fit into? Lobotrichus, which means what? Tuft of, yeah, it's all on one end. It's a tuft of polar flagella. How about this one? This one's got flagella on each end. Amphitrichus. Amphitrichus, they're polar. Amphitrichus has to be polar because they're on each end. How about the cell structure of this? What is, if you saw this under the microscope, how would you describe the cells? It's a diplococcus, yep. Diplococcus. 2 spheres stuck together. How about this one? This is curved, so there's 3 different kinds of curved cells. This is a spirochete. This is a spirochete. And you know what? I happen to know it's a spirochete because that's what it's meant to look like. But if, in real life, if you saw this, if it wasn't moving, you'd probably have a hard time saying it wasn't spirillum. Under the microscope, if you just looked in and saw something like this, you might say spirillum because it looks like it's rigid. How about this with respect to its flagella? Peritrichus, because they're all the way around. Peri means around. Peritrichus flagella. I had another thought about that. But was there one in there? So is there one that's not represented? Monotrichus. One single flagellum. Oh, I know what I was thinking about. The arrangement. So the book has some information about the flagella and spirochetes. So the spirochetes actually have flagella that are underneath the sheath, underneath the surface of the cell. They're called axial filaments. And they don't have flagella that stick out of one end. They have them inside of them that allows them to move through surfaces in sort of a corkscrewing mechanism. So when we stopped last time, we were talking about parts of flagella. We were looking at this figure. One of the cool things about flagella, there's lots of cool things about flagella. You could talk about flagella for a week, but we're not going to. But one of the things about flagella that's really interesting is that bacterial flagella are actually driven by a different electron or energy source than eukaryotic flagella. And if you learn about eukaryotic flagella in Bio 120 or cell biology or any place, you would have learned that the source of energy for eukaryotic flagella is ATP. ATP and the parts of the cytoskeleton that are within the flagella, and they move back and forth against each other at the expense of ATP. But when bacterial flagella move, they move in a very different manner. The whole entire flagellum whips around like a propeller on a boat, and the energy source is proton motive force, or the hydrogen ion gradient across the membrane. So remember, you guys learned about mitochondria. The mitochondria make energy by creating an electron gradient across, or proton gradient across the membrane. They have a whole bunch of protons out here that they generate with proton pumps. They're pumping out here across the membrane, and they build up a high concentration, and they serve as the. What did you guys say something about that? Oh, I know what's going on. Protons. The protons out there serve as the energy source for moving the flagellum. And the basal body actually is a proton pump. The protons go back through. And as the protons go through there, it causes rotation of the flagellum. The flagellum spins round and round based on the fact that the protons get out there. When you talk about physiology and metabolism. We'll talk about more things, how the protons get out there and other things they're used for. That's the energy source for the flagella. So in terms of what they use that for, we said that they use their flagella for moving around. They're moving around and get towards the things that they want, things that they want to swim to. But they can't really steer. So we said the flagellum is like the prop on a boat. If you're driving a boat, what is used for steering? How do you steer it? How many of you have driven a boat? Either a sailboat or a motorboat. They work differently. The rudder, if you have a sailboat, you have a tiller or a rudder that you use to control where the back end is. If you're driving a motorboat, you can move the motor so that affects which direction you're going. You can essentially steer it. But bacteria don't have any mechanism for steering. They only have the mechanism for driving themselves forward with their propeller. So if they want to steer and they want to change direction, they have to stop and then just let the environment, the motion of the stuff that they're floating around in, change their direction, and then they go forward again. So how do you suppose they stop? How would you stop if you were driving a motorboat? There's no brakes. Yes, you have to flip the propeller the other way. You have to put it in reverse really quickly sometimes so that you can stop. And then you eventually you would go in reverse if you let yourself, but if you just want to stop, you just put the propeller in reverse. So that's what's going on here. So the bacteria, when they want to swim forward, they spin their flagella in a counterclockwise direction, which is shown right here. These guys are going. Notice that the direction of the flagella determines which movement occurs. So here it shows it going forward, running when it's going in the counterclockwise direction, and then when it needs to stop, it puts the flagella in the opposite direction and it tumbles. It's called tumbling. So this is standard. This isn't like book vocabulary. People in the flagellum world and the chemotaxis world refer to this as runs and tumbles. So the bacteria go forward when they run. In order to stop, they put their flagella in the opposite direction and they tumble. They reorient randomly in the environment. So this is random reorientation. And then they start the flagella again. They go forward for a while. They tumble. They reorient. They go forward again. They tumble. And they move continuously by this series of runs and tumbles. So that tells you how they move, but that doesn't tell you anything about how they get someplace where they want to be. That is the part that gets to be a little bit interesting. So their movement is based on a series of runs and tumbles. The tumbles result in random direction. So the kicker is, if you are a cell and you need to swim towards the stuff you're getting for food, how is this random runs and tumble, or the random tumbles and the directional runs going to help you? And the book doesn't really do too much, doesn't really help us in understanding that very much at this point, but in order for this to even make sense and be important, we kind of have to talk about what they use this for. And what they use it for is what's called chemotaxis. This is the ability to swim toward chemical attractant. And it's based on a series of runs and tumbles, which gives it the name biased random walk. So like we said before, bacteria sense something that they want to eat, like sugars or amino acids, or something that they need, like light or oxygen, Sometimes it's not chemicals. There's also something called phototaxis, in which they swim into the light because they need the light for energy. I'm just going to talk about this in the context of chemicals. They use what's called a biased random walk. And they have the ability to detect the concentration of chemicals that they're able to sense. So they have molecules on their surface that detect these chemicals, and they have the ability to swim into the region where the chemicals are more concentrated. So I'm going to draw a gradient on here. This red line represents a gradient of a chemical. So this is high concentration down here. Make it red like this. This is low concentration down here. Not very much of it. The bacterial cell is swimming around down here, doing its biased random walk. Here's the cell. It has the ability to measure the concentration of chemicals in its environment, and in response, spend more time in the run part of its motion before it goes into the tumble part again. So if the cell is going in the correct direction, it goes longer. So let's say this cell is going the right way. It goes a long time before it tumbles. Eventually, it does have to tumble. It goes like this. Randomly reorients, and based on just the way that it happens to be set up in the environment, which direction it's facing based on the Brownian movement in the environment, it might be going in a favorable direction. It might be facing the right way, but it's not probably going to be most of the time. Most of the time, it's going to kind of go the wrong way. And if it does go the wrong way, the run is going to be shorter. Runs in an unfavorable direction are shorter. Runs in a favorable direction are longer. Write that down. Runs in an unfavorable direction are shorter. Runs in a longer direction, in a favorable direction, are longer. So this guy's going in an unfavorable direction. Doesn't go very long. Eventually, it tumbles again. If it's lucky, it goes in a favorable direction and goes a long time. It's going, it knows. You're not supposed to anthropomorphize bacteria, but I always do because it kind of helps you learn about it. It has the ability to detect the chemicals, and it goes in a favorable direction. It keeps going before it eventually has to tumble. Reorients. It's going in a favorable direction. It goes a long time. If it's not favorable, it's a short time. Reorients. This little squiggly thing is the cell with its flagella flying apart. If it's lucky, it goes a long time again. So in occasions where it's going in a favorable direction, it goes a long time. So why do you suppose it doesn't just keep going and going and going and never stop? The reason is it has to have the opportunity to reassess the environment. It has to stop from time to time. It has the ability to do short-term memory. This whole thing is based on a bunch of molecular interactions and signal transduction cascades and phospholrelay, transfer of phosphate groups all around, which is all really cool, but we're not going to go into that much detail. We just are going to talk about what they use their flagella for. Why do they want to get around, and how do they manage to sample the environment? So they have receptors on their surfaces that are either bound to attractant. The attractant is stuff they want to get to when it's in high concentration, or when it's in low concentration. Equilibrium favors it going off of the protein, and so it knows how much concentration it has. Do you guys have questions about that? Yeah.
So why does it go in an unfavorable direction?
Why does it go in an unfavorable direction? Well, it doesn't. The reason it goes in that direction to begin with is that when it stops moving, the effect of the solution that it's in pushes it into a different direction. Because if a bacterium stops swimming and it doesn't have anything pushing it, either waves or anything, it's just stationary, Brownian motion forces the front end of the bacteria to move around. It doesn't really have any control over which direction it's going. So when it pauses, I mean, if everything, if all the molecules were completely still, which it can never be, it wouldn't move, it would just keep going. But the force of the environment has to move a little bit, and that's why it ends up unfortunately going in the wrong direction. I mean, every once in a while, it probably gets lucky and doesn't go too far out. As long as it goes, if it goes in an unfavorable direction, it corrects itself sooner than if it's going in a favorable direction. So eventually, they find their way to the food source that they're looking for, the light intensity that's most favorable to them. Yeah.
So you said the flagella have chemical receptors?
Well, it's not the flagella that do, but it's the front end of the bacteria. So the flagella are on one end, and And down here they have proteins that stick through the plasma membrane that actually bind to chemicals.
And then, even though there's chemical receptors, it's the environment that's pushing them along.
Is the environment what?
Is the environment that's pushing them along in this direction, or is it the chemical receptors?
It's the chemical, it's the flagella that push them. It's the chemical receptors that are able to tell whether they're going in a favorable direction. The receptors are what help detect its favorable direction. And then there's a whole bunch of stuff in between the front end of the bacteria where the receptors are and where the flagella are that sends signal through the cell to control how long it stays going in that favorable direction. Any other questions about that? Okay, so let's talk about, so remember we're doing, this is about cell biology bacteria. We started on the outside of the cell, talked about structures outside, the glycocalyx, which is a sugar polymer. We also said sometimes it's amino acids. I said that, we should write it down. It's kind of annoying because it's called a glycocalyx. You think it would always be sugar. The capsule specifically is sometimes amino acid. Bacillus anthracis has a protein capsule. That's a really important organism. We talked about the slime layer. We talked about, somewhere I just saw that I wrote on the virulence factors. Why is the capsule a virulence factor? Two things that make it a virulence factor. One is that it's involved in sticking to things, which is important for its ability to associate with the host. What was the other one? Yep, protects it from phagocytosis, so that our white blood cells can't gobble it up and kill it. Alright, so we talked about those things. We talked about flagella. Next thing on the outside of the cell. Fimbriae and pili. So fimbriae and pili, these two words are sometimes used interchangeably. The fimbriae and the pili, depending on where you went to school and what part of the world you grew up in. Usually we call it fimbriae. This part of the country, this part of the world. These are surface structures. They're little hair-like extensions on the surface of a bacterial cell that allow it to stick to things. Allow bacteria to bind to surfaces and colonize. So there's a figure in your book that shows falsely colored electron micrograph. It's really pretty. This, what shape of cell is that? It's kind of squashed and deformed because it's been treated with. This is actually E. coli. It's a little bit elongated and not very cottage shaped. These yellow things around the surface are the fimbriae. And they're much shorter than flagella. They're thinner than flagella. They the main function of sticking to surfaces. So what is meant by colonizing surfaces? So one thing is if a bacterium sticks to a surface. Another thing is if it colonizes. If it actually sticks to it and then colonizes, that means that it attaches there and grows and stays around. It's like pioneers out on the prairie. If they colonize, they actually stay there and set up a community where they live for a while. So why do you suppose we care about them? What kind of surfaces would they ever stick to that we would care about? Your lung, your large intestines, your teeth maybe. So these are also virulence factors because they're involved with the ability of bacteria to stick to the host. Virulence factors, remember, are the parts, properties, and what's the other P? Products. Parts, properties, and products. This would fall into the category of a part of a bacterial cell that's involved in causing disease. The important thing about them is that they are very specific for their target. So their target might be some kind of slimy stuff on the bottom of a boat, or it might be some protein that accreted to the bottom of the bathtub. But they're specific for those places. It's oftentimes some kind of protein on the respiratory tract. So that makes these things virulence factors too. Very specific for target. And they also control what kinds of surfaces different kinds of bacteria can stick to. So what does that remind you of? There's an analogous thing on another thing we talked about. It's like the spikes. They're sort of like the spikes. They're also very specific, responsible for getting the virus to stick to the target cell. See, they don't show. Okay, that's all we're going to say about the detail of that. So as I said before that fimbriae and villi are used interchangeably, but there's one case where villi is always used separate from the fimbriae, and that is what's called the sex villi. Sex villi are used for cell contact during transfer of DNA. So pili, I didn't say this before, but pili is plural. Pilus is singular. So bacteria, when we get to the section on bacterial genetics, let's see here. Chapter 8, we're going to talk a little bit more about how different kinds of bacteria are able to get DNA. They get different kinds of DNA, and one of the ways that they get it is by conjugation. Transfer of DNA during conjugation. Which is a process by which one bacterium gives its DNA to another bacterium. It's also known as horizontal gene transfer. So I'm going to show you a picture from chapter 8, which we're going to talk about later, that shows this conjugation going on. Here's two cells. These happen to be two cells of the same species. The F plus cell, the F minus cell. You can see this thing has fimbriae all the way around it. You see those little hair-like appendages? Those are the fimbriae. This is the sex pilus, which is literally a hollow tube that attaches one cell to another. And when the cells are conjugating or mating, DNA passes through this tube from one cell to another. So there's some detail on that we'll talk about in the next chapter. I just want to show you briefly so you know why this is important. Here's a plasmid. A piece of DNA in this cell gets transferred across the sex pilus, and then this cell gets that piece of DNA. So is that a very big deal? Why would that be noteworthy that one cell is giving DNA to another like that? Would it help colonize? Good, depending on what the function was in there. What kinds of capabilities would a bacterial cell get its DNA that we would care about? Antibiotic resistance. This is the way that antibiotic resistance can spread from one cell to another. So this cell could have genes for antibiotic resistance in there. It could pass it on to its neighboring cells. So it could pass it on to its progeny through vertical gene transfer. It could pass it on to neighboring cells through horizontal gene transfer. And these neighboring cells don't even have to be the same species. There are bacteria of different species that can mate with each other and share their genes with bacteria that aren't even of the same species, which would be a really big deal if it's antibiotic resistance. It can spread around from species to species that way. So we'll talk more in detail about that when we talk about genetics. So let's talk, okay, so that's highlights of the outside of bacterial cell. Moving inside the cell, the border, before you get inside, is the bacterial cell wall. So this is a really important part of cell walls. And some microbiology books call this the most important part of the bacterial cell, which I think is kind of funny, because how could you say that there's just one most important part and not say it's the DNA, or not say it's something that's essential to being alive? You can't really say one is most important, but the fact that some authors say that it is tells you that it is really super important, because it provides strength to the bacterial cell. It provides strength, which is really important to single-celled organisms. I'm putting single-celled organisms in parentheses to remind you that we don't have a cell wall, and we don't really have problems with providing strength to our cells. But if you think about the way bacteria live, as single-celled organisms swimming around in the world, they are swimming around in an environment that is osmotically different from their own cytoplasm. And that is the reason why they have to have a way to deal with, to provide strength to them. So this is a bacterial cell. Actually, there's in the book, it is this figure I put on the Moodle. It shows a little bit about the cell, the environment. It's on page 92. If you imagine a bacterial cell swimming around in the environment, would you imagine that the environment is isotonic to the bacterial cytoplasm, hypotonic to the bacterial cytoplasm, or hypertonic? So what is the difference between isotonic, hypertonic, and hypotonic? Isotonic means that it's the same concentration of solute on the inside as the outside. Hypertonic means that the inside of the cell is more concentrated solute, saltier, or it's more full of protein. If the inside of this cell is hypotonic relative to the environment, it means that it's less salty. The environment is more salty, right? So would you guess that the inside of the cell is more concentrated or less concentrated than the environment? It's more concentrated. This is way more concentrated because it's jam-packed full of proteins and ions and nucleic acids. And the bacterium swimming around out in the environment. And the reason that this used to seem kind of counterintuitive to me is the stuff that we grow bacteria in the lab is so turbid, it's not turbid, but it's so concentrated with protein that it's actually yellow. But that's nothing compared to the inside of the cell. It's probably viscous with protein. And so if the cell didn't have a cell wall, what would be the outcome? So this is where the figure in the book comes in handy. If you have cells in a hypotonic solution, which is shown right here, water moves into the cell. If the cell wall is strong, it contains the swelling. So why would swelling occur? If water moves into the cell, it has a tendency to move into the cell in order to dilute the inside of the cell. And if that happens, the cell would swell. And if it didn't have a cell wall, the only thing it would have to protect is the plasma membrane, which is really fragile. It's not really strong in terms of this kind of a force. It would cause the cell to blow up and all the contents to ice out of there. So what the bacterial cells have to have is a network around the outside that gives them strength, and that network is called the cell wall. It gives them protection from osmotic forces. So it's important for the cell to provide strength. It's also important in terms of, it's important clinically and in terms of pathogenicity and virulence. So it's important for clinical virulence concerns. And one of the reasons for that is that the cell wall is the site at which most of the antibiotics that we have work a target. It's the site that most of our antibiotics work. The bacteria have a unique cell wall in terms of its chemical composition and the way it gets put together. It's something that we don't have in our bodies, and so it's something that we can effectively target with bacteria to prevent them from causing infection. You can use antibiotics against this structure because we don't have that structure. If you can block the formation of that structure, you're targeting bacteria without affecting the human host. So we're going to talk about what that structure is next time. And if you guys want to be a step ahead, it's always a good idea. Now look at these pages. Page 84. 84, this figure. 85 shows how cell wall is constructed. And it continues on to page 86. So we're going to differentiate between two different kinds of cell wall, the gram-positive cell wall and the gram-negative cell wall. So you guys can read ahead for next time on that stuff. Does anybody have any questions? We have another minute. 1 minute is 2% of our whole time. Do you have an announcement about that?
Tomorrow we're doing grant things.
Oh,
Don't
perfect.
forget to,
Well, it would have been perfect if I would have got it today.
we'll start it tomorrow.
Yeah, you'll start it tomorrow and
Print
then
out
we will.
the